Today, we're going to learn about action potential. Demonstrated before you, we have a cell membrane with three gates, represented by the different colored toothpicks. The M gate and the H gate, the blue and the red, make up the sodium channel, which passes sodium, or the black beans, between the outside and inside of the cell. The plain toothpick, or the N gate, makes up the potassium channel, which passes the red beans, or the potassium ions, between the outside and inside as well. We also have white beans and pasta spirals, which make up the chloride, which are the beans, and proteins and DNA, which is the pasta. Differences in concentrations of ions between the inside and outside of the cell leads to differences in charges, which we call the resting membrane potential. In our example, you'll see that inside the cell we have a negative 9 volts, and outside we have 7 volts. This gives us a resting potential of 16 volts. Now, a stimulus is introduced to the cell, causing the M gate to swing wide open and allowing sodium ions to diffuse into the cell.
Now, this shift in sodium ions is called the depolarization process and leads to a different potential across the cell membrane. The depolarization process then causes the voltage-gated potassium channel, or N gate, to open, and then the sodium channels to close. This allows the potassium ions, or red beans, to move from inside to outside the membrane. As you can see, the repolarization process has re-established the original membrane potential. However, even though the original membrane potential has been restored, there has been a reversal in the concentration of sodium and potassium ions. So, in order for the ions to go back to their original places, the sodium-potassium pump comes into play, where ATP is used to restore the original concentrations.